Hello children, today we are discussing about an activity, activity 1, present of starch, present of starch, a type of carbohydrates in leaves. You know the steps of a lab activity or activity. So aim, material required, precautions, procedure, observation, conclusion. And these are the steps we need to follow while doing an activity in the classroom or in the laboratory. So here in activity 1 he is telling us, and here we are doing the activity to know the presence of starch in leaf. So each and every activity, activity is having its own aim. So aim of these activities to know the presence of starch in leaves, to study the presence of starch in leaves. Again, second step, material required. So what are the materials required to do this activity? We need to know. So here you see a glass beaker, a test tube, a tripod stand, a Bunsen burner, methylated spirit, water, a thin and soft leaf which is well exposed to sunlight, and iodine solution, petri dish. In all these are the materials we required for to do this activity. Next, and what are the precautions we need to take while doing this activity? So here you should not, we should not touch the iodine solution, and we should carefully pour 1 or 2 drops of what that is iodine on the leaf. So here these are the precautions we need to take while doing this activity. And now I am telling the procedure. First, we need to take, we need to pluck a leaf, a soft and thin, well exposed to sunlight leaf, and that leaf we need to keep in a test tube, and the test tube is having what methyl spirit, and that test tube we need to place a beaker which is the 3 by 4th of the beaker is with water, and we need to keep it on a asbestos sheet above the tripod stand, and we need to boil or heat the beaker with water or test tube with methyl spirit and leaf with a burner. And after some time you will observe the green leaf turn into white. It loses its green color structure, and the chlorophylls are dissolved in the methyl spirit. And that leaf, the discolored leaf, we need to take it out with the help of a brush, and we need to keep it on the petri dish. And after some time we need to add 1 or 2 drops of iodine on the leaf. So here what is the observation, what are the observation you will find while pouring 1 or 2 drops of iodine on the pale color leaf, discolored leaf. Here leaf turn into bluish black, and that tells that leaf is having starch. And this activity is telling us that, confirming that leaf is having was starch, and this is the test we need to know the presence of starch in the leaf. So in this way we can find, we can observe, we can see, we can find out the presence of starch in the leaf. And this activity is doing, we are doing for to know the presence of starch in the leaves. So here now I am asking a few questions. What are the apparatus, what are the materials we require to do this activity, to do the study of presence of starch in the leaf? Second question, name the chemical which we need to use, which we need to take in the test tube. Third question, name the reagent which is used to test the presence of starch in the leaf. So here these are the questions you need to write, and you need to write all the steps of the activity. First one, aim, material required, precautions, procedure, observations, conclusion. So in this way, we need to do this activity. So this is the activity 1, and figure number 2a, boiling the leaf, and figure number 2b, iron test. Thank you.